Research webinar. This webinar is being offered as part of the Forests in a Dynamic World webinar series hosted by the Delaware Forest Service and the Delaware Department of Agriculture and the Delaware Coastal Training Program, which is part of the Delaware National Estuarine Research Reserve and the Delaware Department of Natural Resources and Environmental Control. Good morning, everyone. My name is Jennifer Holmes, and I am the Coastal Training Program Coordinator for the DNIR. Just a few webinar hints and tips. Please keep yourselves on mute during the presentation. This will help reduce any background noise, and feel free to turn your cameras on or off by looking at the bottom portion of your screen. There's a camera icon. If you're having bandwidth issues this morning, you may want to leave your camera off. You can also mute and unmute yourselves by clicking on the microphone icon also at the bottom of the page. We encourage you to use the chat box today to ask questions. The chat box looks like a thought bubble and can be located at the bottom right of your screen. If you have any technical difficulties this morning, please send a private chat message in the chat box to Kristen Thornton, and she will be able to help you. Just to note that the webinar is being recorded today, and a link to the webinar will be provided in a follow-up email. We are also pleased to have with us today representatives from the Forest Service, and I know Mike Valente is on today with who is the Forestry Administrator. So welcome to our forestry partners. We are also pleased to have with us today our panel, and we have Dr. Tara Trammell, the John Bartram Assistant Professor of Urban Forestry at the University of Delaware, Dr. Stephanie Stotts, an Associate Professor of Environmental Studies at Wesley College, and Lizzie Powell, a PhD student at the University of Maryland and a Margaret A. Davidson Fellow for the Delaware National Estuarine Research Reserve. At this time, it's my pleasure to hand it over to Dr. Trammell for her presentation, Small Forests Embedded in Developed Landscapes, Environmental Stresses, and Ecosystem Services. Tara, I'm going to stop sharing so that you can share your screen. Welcome.
Thank you. I'm very happy to be here today to talk with you all about some of the research that's going on in my lab. So can everyone, is that good? So in my lab, we study how altered urban environmental conditions, especially in our cities and towns, influence small forests, and how this in turn can influence how these forests provide ecosystem services or ecosystem benefits. So we know that cities are where we have our concentrated, human population globally. The cities are also hotspots for energy use and emissions, which can have global implications on our environment. So 25% of the world's population resides in the United States, Europe, and China, and this is also where our temperate deciduous forest biome is located. So globally, the temperate deciduous forests are located where we have these densely populated regions. So some of my colleagues did a spatial analysis looking at all the temperate deciduous forests across the United States, and what they found was that 94%, or the majority of all of our forests, are small. They're less than 50 hectares. So the majority of our forests are small. They're located in densely populated regions where concentrated human activities can really alter our environment. So I'm going to talk about some of the really well-documented sort of abiotic phenomena that we see in our cities. The first one is the urban heat island effect. And this is where the temperatures that are in our city centers can be 1 to 2 degrees C warmer than the nearby rural environments. And depending on the biome that you're in or the time of the year, sometimes that temperature difference can be up to 9 degrees C difference. We also have documented very clearly what is called an urban CO2 dome effect. And this is where our cities have greater concentrations of carbon dioxide in the atmosphere. And this can be up to 200 parts per million greater in our cities. But in addition to these abiotic factors, densely populated regions are a point of entry for non-native species, and a lot of these species can become invasive. These invasive species then have detrimental impacts on our ecosystems, such as reducing native species and altering the species composition. So it's important to study how these forests can withstand all of these abiotic and biotic sort of changes in our environment, yet still maintain ecosystem structure and function, or ecosystem services. So the Millennium Ecosystem Assessment divided ecosystem services into 4 categories based on the type of ecosystem service. So a lot of focus has been on the provisioning, regulating, and cultural services that our ecosystems provide. But all of those depend on the supporting ecosystem services underlying these. So services such as biodiversity, nutrient cycling, and primary productivity, or the total amount of plant growth. So studying these ecosystem services can provide an indication of how our forests are functioning in the face of environmental change. So we work in we can conduct our research in a long-term urban forest network called the FRAME. And we have 38 forest sites in our region that experience a wide variation of these urban abiotic and biotic changes. So our forests vary in the amount of urbanization intensity around them. So we have forests that are surrounded by rural landscapes, forests that are surrounded by suburbs, and forests that are surrounded by dense urbanization. So studying these small forests across this gradient can provide this indication of how the forest is responding to abiotic environmental changes. Our forests also have a gradient of non-native invasive plants within them. So this is showing the digitized or aerial coverage of our most dominant invader, which is multiflora rose. And we have sites that have almost no invasion up to sites that are almost completely covered by invasive plants. So studying our forests across this gradient of invasion can provide an indication of how the forests are responding to these changes in species and other biotic factors associated with invasion. So because our cities a lot of times are points of entry for native species, we expected to see a relationship between the amount of urbanization surrounding our forests and the amount of invasion within them. So on the x-axis, we have the urbanization gradient, and on the y is our invasion gradient. And what we found was that there was no relationship at all between these two factors in our forests. So this means that we can look at these two factors and how they're influencing our forests separately. And I'm going to first talk about how they influence the structure of our forests. So it's important to talk about forest plant biodiversity because it's a determinant of how our ecosystems are functioning. But diversity is also really important for thinking about resilience to pests or pathogen outbreaks or other sort of extreme weather events. So I'll first talk about the canopy trees in our forests. And what we found was that across all of our forests, the majority of our trees, so over 96% of all the trees we sampled are native, which is a good thing to find. And then to assess the relationships between the diversity of our tree species and invasion and urban gradient, we did scatter plots to look at that. So in the figure on the left, we're looking at the invasion gradient on the x-axis, and the figure on the right is the urbanization gradient. And then our y-axis is the number of tree species we find within each plot. So we found that the tree species richness increases with urbanization. And so we wanted to know if this was being driven by more non-native plants within that. So we divided this into looking at the origin of the species, whether they were native or non-native. And what we found was that native trees increase and non-native trees increase. So we saw that it's both that are increasing in our forests that are surrounded by more urbanization. So this is an interesting result, and it shows us that these forests that have a lot of human activity around them can still maintain native species. So then we looked at the understory and we saw something very different. So almost 60% of an understory is made up of non-native invasive plants. And we wanted to look at the relationship between the diversity of the understory and our invasion and urban gradient. And what we found was that the number of species within the understory increases with both invasion and urbanization. So if we divide this by looking at the origin of the species, we see that, of course, it's not surprising that with increasing urbanization within it, we have more non-natives, but that definitely clarifies that it's the non-native plants. But what's interesting about this is that it shows that these non-native plants, these non-native invaders can co-occur, and that we're not necessarily seeing one dominate over others. With the urbanization gradient, what we saw is that this was increasing native species in the understory. And this is a very interesting result and a good result, but it also supports that idea that our forests with a lot of human activity around them can still maintain native species within them. So we want to see how this relates to how the forests are functioning. So for today, I'm going to talk about the potential for forest carbon storage in these small forests. And it's really important because forests globally are an important sink for carbon. So I'm going to show here just a sort of simple conceptual model that shows the relative sort of size or importance of two different ecosystem processes that control carbon storage potential in our forests. The first is the production or the amount of biomass the trees are putting on. So the production is shown in the top in the green box. And then how fast, so this is the organic matter inputs we have going into the forest soil, and then how fast those organic matter inputs decay. So the two processes and their relative sort of size to each other is really important in determining the amount of carbon that can be stored within our forests. So to do this, we looked at the leaf biomass that was being produced by our trees each year as an indicator of productivity. And then to look at an indicator of our decay rates, we used a uniform substrate. And so what we found is that in our forests that are surrounded by rural landscapes and they're not invaded, that we see what you typically see in a sort of a healthy forest is that you have a lot of productivity happening in the trees, and you still have good decomposition happening to release nutrients, but there's more production happening than decay. And usually this then leads to soil carbon storage in the system. But what we found when these rural forests are invaded is that the productivity decreases and the decay increases. So this leads to less soil carbon storage potential in these forests. And that's because you have less organic matter moving into the soil, and then when it gets there, it decays a lot faster. But when we take these invaded forests and put them within an urban context, we saw an interesting trend that the production goes up, more similar to our rural uninvaded forests, and the decay slows down. So this can lead to more soil carbon storage potential in these forests. And that's what we found, was that there was greater soil carbon and nitrogen in our urban invaded forests relative to our rural invaded forests. So this suggests that there's something about the urban condition around these forests that can potentially offset those negative impacts of invasion. So just to sort of recap what we saw in the forest structure and function of these small forests, overall we saw optimism that the forests that are surrounded by more human activity or more urbanization can maintain native species. These are not degraded ecosystems. And where we see changes in the system is more driven by the invasion. With the ecosystem function, the productivity, and the carbon storage, we saw that our invaded forests have lower biomass production, faster decay, which leads to less soil carbon storage. And we also saw that something about the urbanness of the system can help offset these sort of negative impacts we typically see of invasion on soil carbon storage potential. Now, I didn't get into the details about this today, but we do study nitrogen cycling in our systems as well. And we found that urban forests have various sort of characteristics that have made them able to act more like a nitrogen sink, so to store more nitrogen. Whereas forests that are invaded have, the characteristics or these fast production rates of nitrogen in the soil that can lead to more nitrogen coming out of these systems. So overall, we saw that, these forests that are surrounded by a lot of human activity can support these, ecosystem services, especially the supporting ecosystem services that are really important for ecosystem function and structure. But the invasion appears to be a threat to our biodiversity, our carbon storage, as well as the sink potential for nitrogen in these systems. So it's really important then to approach these forests with a management perspective for invasion. So my PhD student was really interested in doing an experimental active management project for his PhD, looking at invasion and doing some treatments to help boost the native species and improve soil nitrogen cycling rate. So we conducted our manipulated experiment. The goals were to eliminate the plant invaders in the understory and restore our native plants. We did this in 3 different forests, so forests with different levels of invasion intensity, to try to understand how the current level of invasion in the forest could influence the success of our management practices. So we did a mechanical removal of all the non-native invasive plants in the understory. This is shown by the inner green box. I don't know if you can see. And this diagram. the lighter green. And then we did several different treatments. So we had control treatments where we didn't do anything outside of our removal zone. We had our removal zone, and then our treatment plots are shown in 1 here, the ones. This is where we removed the invasive plants. The treatment plots 2 are where we removed the invasive plants, but then we added a native seed mix to the forest floor. And then the treatment plots in 3 had the invasive plant removal, the native seed mix, and then a carbon-rich soil amendment added to that. So in this figure, what we're looking at is called rank abundance curve. So basically, this is showing the different species and their abundance across our sites. So in the right or on the left-hand side, we have the low invasion site. In the medium, we have, or in the middle, we have the medium invasion site, and on the left-hand side, we have the high invasion site. What we're looking at here is the woody plant community in the understory. And the 3 different lines and the 3 different colors represent, in red, it's 2017, so this was before we did any treatment. And then the 2 years following our treatments in 2018 and 2019 are shown in blue and green. So these are what we saw in our control plots where we didn't do anything. And not surprising, our most abundant invader remains the most abundant, which is what we expected. But what was interesting is that we were seeing that native species were becoming less abundant across these forests over time in just a couple of years. So in our low invasion site, we saw oak seedlings dropping out, and in our high invasion site, we were seeing less spice bush, less abundance. And we also saw this pattern in the other sort of layers of the understory, in the herbaceous layer and in the ground cover as well. And we actually saw new invasives coming into the ground cover layer. Now this graph is showing our, at the end of our experiment, the change in the native species richness over the time period. So we have the low invasion on the left again, the medium invasion in the middle, the high invasion on the right. The top 3 panels are showing that mean native species richness, so the change in that over time. And on the bottom, we're looking at the floristic quality of the plant community and the change in that over time. So in general, what we saw was that we had increases in native species richness, and we had increases in floristic quality. Now, how the actual plant community is responding and which treatments were most effective in these increases changed depending on which kind of forest we were in. So my PhD student is in the process of writing up his results, and so we're hoping to have some more sort of potential guidelines for managers and when some of these treatments might be more successful than others. So overall, the management increased the native species, it decreased the exotic species in our understory, and increased the floristic quality. So in my lab, we're also building, working on more restoration efforts that are focused on, in these degraded forests that are highly invaded, some treatments that we can do to try to help restore native plants. And we're also working in forests that are experiencing really reduced oak regeneration. So there'll be more to come on some of our restoration efforts in the future. So I just want to thank my funding sources, my collaborators at the Frame, and the lab members, my lab members, especially my PhD student who did all the experimental work, Eric Moore, and I would be happy to take any questions.
Great, thank you so much. That was extremely informative. We do have a question in the chat box. The question is, how do you account for the role of deer herbivory in your assessment? This person says that they used to live in a forested area in southern Chester County that had a high enough deer population that the understory was almost non-existent except for multiflora rose.
Yeah, so that's a really important question. And we definitely have a lot of deer and high deer densities in this area. So this particular research has always been in forests that don't have deer exclosures. So we're trying to, because deer are such an important part of the dynamics in our plant communities, and not very many people have the, maybe the resources to put up deer exclosures to bring back the understory. So we're always trying to sort of understand what is the sort of species composition in these forests with that sort of part of the, equation as the deer herbivory. So this is all including that as part of it, as well as when we're doing these management treatments, they were not protected from deer. So we saw these increases in the native species richness, even in forests that were able to have deer come in and influence that. because, you know, we were trying with, especially with the experimental work, we were calling this easy to implement and cost effective. So the mulching treatment was basically mulching all the stems that we cut down. So we just had to take a wood chipper into the forest and chop it up and then spread it. And the seed mix was easy to spread. So we were trying to come up with things that managers could do after removing the invasive that would help reduce what they call secondary invasion. A lot of times you take out invasives, the next thing that happens is more invasives come in. So what could we do to sort of try to reduce that from happening? In our median invasion site, we did see a lot of invaders come in still after, but we also saw the native species increasing at the same time. We didn't see the invaders coming into the high and low. So it depends on the sort of level of invasion you already have as to how these treatments might sort of influence that post, you know, removal and treatment plant community. But yeah, deer are really important. For some of the future work that I'm doing on oak regeneration, we are going to use deer exposures because it's really hard sometimes to get these things to happen without that. But yeah, so we have multiple sort of methods that we use that try to take that into account.
Great, thank you so much. Yes, I've seen a lot of herbivory by deer in some of our restoration sites as well.
Yeah.
How do you account for, so another question, how do you account for your statement that native species richness increase with urbanization?
Yeah, so, you know, some people have asked whether it is due to the fact that a lot of times settlements happen where there's more diverse biota, typically anyway. But we're working at a scale that has not been measured before. So there hasn't really been research showing what is that sort of influence of urbanization around our forests and the amount of the natives within that. And so at the scale that we're working at, the biodiversity is similar across all these sites, or the potential for it is. So the fact that we're seeing that within these urban forests is a positive result. Part of this has to do with land use history. So I didn't have time to get into that, but where we see the most invasion are in what we call our younger forests. So these are the forests that have had, we did aerial imagery analysis looking at the time since the canopy closure, because all of these are second growth forests. At some point, they were something else, or they were logged at some point. So we went back to 1937 with the first aerial imagery that's available, and looked at that, you know, time since canopy closure. And that's really where you see the most invasion is in our newer sites, which isn't too surprising when you think about it. You know, there's a lot of light coming, there's more light coming in, and they've had a chance, I think, depending on the time period. So it also has to do with when these forests, the canopy closed. It's the same time that these plants were in the system. They were around, they were dispersing, and they were able to get in and sort of, you know, become widespread. So our urban forests, a lot of times, they've been forests longer because they either were in locations people didn't want to develop, or they were just something that they wanted to protect because it was a park, or it was something that was already being utilized as forest. So it has a lot to do with sometimes with land use history and just where they are in the landscape too.
So just as a follow-up to that, did you have any old growth plots? Or could you even find them in the urban areas? I don't know.
Yeah,
going.
we definitely have forests that were solid, nice, beautiful forests in 1937. But what happened before that is a little harder to tell. We did have a landscape architecture grad student work with us for a while and trying to build more complete and detailed land use histories of all of our sites. But it's a really long sort of painstaking process, and we didn't get through all of it. So yeah, some of our forests have been forests for a really long time. But how long is hard to say. But we've definitely had some that were much older than 1937, but how far?
And so I guess there was a question that is, okay, thanks, Blake. I just saw that. Okay. Another question was, what size were the different experimental plots? I think that you had it on the slide, but I didn't know if you size.
Let me see if I can go back.
Or I don't know if that was.
Yeah, so oh, is somebody talking? I was talking. Yeah, so this sort of lighter green area here with the removal zone was a 20 by 20. And then, so our actual treatment plots were smaller. They were like 2 by 2 meter plots within that. And then they kind of had to be scattered around the trees. That's what these little green dots are supposed to represent, trees. So we had to kind of move them around depending on that. That was
Great,
the
that was-
question, right?
Yep, that was helpful. Thank you. And then the last question for now is, did you perform any kind of landscape ecological analysis looking at shapes of patches or patterns, etc.?
Yeah, so we've done quite a bit of GIS spatial analysis on our forest. So we have the size of the forest patch, we have the edge to interior ratio, and we've used that in sort of analyzing the more complex patterns of our species composition. And so we do have a tendency for our urban forest to sometimes be smaller than the rural forest, but it's not a really strong relationship. But sometimes some of these factors are hard to separate. So we definitely see the urban forest could have be smaller, but our edge to interior ratios really weren't driving a lot of our plant community sort of changes in the composition. It was really being driven by the invasion in the understory.
Great, thank you so much. That was helpful. We are needing to get on to our next presenter today, but thank you so much, Tara. We really appreciate that. If I know there's another question in the chat box, and we will try to get to that. Either answer it. Feel free, Tara, to answer the chat box if you'd like. At this time, I'm going to hand it over to Dr. Stotz, who is going to give us a presentation on when did the baby trees stop growing, using tree ring analysis to date tree regeneration in a transgressing coastal forest. Stephanie.
All right, let me share my screen here, pull up my presentation. Okay. Good morning. That was a really interesting presentation with so much information. So I'm going to be presenting on some work that we're doing with this coastal forest transgression, which is the inland migration of salt marsh into coastal forests. I am an associate professor of environmental science at Wesley College, and I also run a tree ring laboratory. So I'm really interested in using annual tree rings to figure out what the environment is, a tool to look at the history, the past environmental conditions. And on this slide, I have two photographs. The first photograph is a coastal forest, and it is experiencing some stress because you can see there's a lot of holes in the canopy, a lot of light getting through, but we can also see that there is still regeneration because you can see the baby trees underneath. So this forest is not overly stressed. It's still holding on because we still have regeneration. The other photograph is a picture of a ghost forest that is located along the St. Jones River in Kent County, Delaware. And this is a system that has fully transitioned. It is no longer a forest, it is now a salt marsh. And this concept of ghost forest has become really popular in recent years. About every news media source there's been has run a story on it, from local news sources like the News Journal to NPR and the New York Times and Time Magazine. And I think they've really captured people's attention because it's this visualization that people can see that the landscape has changed, that it is no longer a forest, it has changed to something else, and you can't deny that that change has happened. The rest of these images, well, the next couple slides are images from a collaborator, Karen Geeden from George Washington University, and she has a really nice story map that goes through the process of how ghost forests form. The first thing is that when the trees become stressed, they are going to slow down their growth, and also their sap flow is going to slow down. And in this photo, you can see these little sap flow sensors, and they will measure that sap flow, and you can compare that to different environmental conditions, things that are happening. The next step that we see in this formation process is that any young trees that exist become stressed and die, and you can kind of see in these pictures that you've got the taller trees and then you've got these dying, unhealthy small trees. And also your tree regeneration, your germination just stops. And you end up with these coastal forests that are dominated by old species, by old trees, not old species. And then with the stress in the opening in the canopy, the more light, you start to get invasions. It may be salt tolerant species and grasses that move in, or in a fresher system, you tend to see invasive species move in. We tend to see a lot Phragmites moving in the understory of the system. And then eventually the trees die, and leaving a ghost forest, a lot of these standing dead trees in the middle of the marsh. I am involved in a project, I'm a collaborator, that is trying to look at these processes and what drives this transition. So are these slow processes driving this transition? Is it that your water table is rising and the trees are getting stressed simply because the water table is so high? Is the water table becoming salty with higher sea levels? Is it these slow processes, or is it that because the sea level is higher, when a storm comes, that storm surge comes further inland and it's bringing salt, sort of this really fast and rapid process? This overall project is called the Coastal Critical Zone, and it's an NSF-funded project through the Critical Zone Network. And I put the website for this project on this slide here. The two overarching goals, which are to investigate those drivers that I just spoke about, the slow and fast processes, and what is it that is really driving this transition, this inland transition. And then the other step in this is looking at the feedbacks. So how does this process change nutrient cycling? And as these trees die and their roots decompose, does the land surface drop down, allowing the salt marsh to move in more rapidly? So looking at these different feedback processes. We have 3 study sites. One is Monet Bay, which is part of the Maryland NEAR system, the VCR, and we're still working at trying to find our Delaware study site. One of the sites that we've considered, which probably most people are very familiar with, is the Delaware National Estuarine Research Reserve, the St. John's Reserve. And there is evidence of some forest retreat in this system. The problem is that we're finding most of our coastal forests in Delaware are very narrow. So it's hard to really pinpoint that the stress is definitely coming from the salt when you have such a narrow forest. It may be coming from the upland side as well. And so our requirements are that we have this transition. We want to have a salt marsh. We want to have this sort of ghost forest. We want to have a transitional forest happening, a low forest and a high forest. And we would like that this be a relatively wide forest. So if anybody is aware of sites in Delaware where we have a relatively wide coastal forest intact with the obvious signs of this transition occurring, I would be very interested in hearing that. We are going to go out and look at a couple of places in Sussex County next week to try to scout out sites. But for the rest of the presentation, I'm going to talk about kind of a pilot project that has been a part of this overarching project. And that's occurring in Monet Bay, which is on the eastern shore of the Delmarva Peninsula, and it is just west of Princess Anne, Maryland. And this is the study site setup. I just realized I don't have a scale bar on here. So each one of these transects is about 300 meters long. And you can see here how we have it set up, that you've got marsh study sites, you've got the transitional forest, you have low forest, and you have high forest. And within these transects, because there are a lot of different research teams working on this, there are sediment elevation tables, there are sat flow meters, there are groundwater monitoring wells, there are ISCO samplers to measure the what's running off of this system, to really try to understand the project as a whole. The research question that I am investigating in particular, though, is trying to figure out in the system, when did this regeneration stop? We looked at the process and the steps, and we know that regeneration stopping is one of the early steps in this inland transition. And so if we can pinpoint the timing of when it stopped, we can look at environmental conditions, storms, things that were happening at that time to try to investigate what really is driving that process. We set up several study plots. These plots are about 50 by 50 meters, and they range from the low forest. In the low forest is when we start to see regeneration. So they range from the low forest up into that ghost forest margin. We sampled every specimen that we could get within each of those study plots. We used an increment borer, and we tried to go all the way through the tree just to get a duplicate sample. That wasn't always possible depending on the size. We were focusing on Pinus taeda, or loblolly pine, as it is the dominant species in both the VCR and the Monet Bay sites. Once we collected these cores, we glue them into a core mount, and we sand them down with progressively finer sandpaper until they get really smooth and you can see the rings really clearly. This is just a photograph from inside the microscope. And the lighter wood would be your early wood. It forms in the spring. The darker bands are your late wood, which are going to form in the summer into early fall. We have gone out at Monet Bay. We've collected samples from 83 trees. Half of those were collected in December, and then the other half were just collected last Friday. At Wesley, we work with undergraduate students, and so we need to have kind of these smaller bites of projects that undergraduate students can tackle in a year or in a semester and still have something to present. Within our lab, we have two different techniques for how we can measure tree rings. One of this is a manual measurement. So you use a linear encoder, a microscope, and you're just manually, individually measuring each tree ring. We also have an automated system where you scan in your cores, and it's called Windendro, and it will automatically measure your tree rings. But our question was, that the undergrads undertook, was which one of these methods will work in our system? And in early research, early data that the undergrads came up with, they found that in some cases, the automated system worked really well. The automated system would be the preference because it's much faster and easier to use. However, some trees are, the manual system is definitely going to be more accurate. With the manual system, you can see the individual cells, which allows you to really be able to detect missing rings and false rings and rings that are really, really tiny. The disadvantage to the manual method, though, is that it is very tedious. It's very labor intensive. And so trying to figure out which system will work for this project. They were able to this semester analyze about 25 tree rings. And for many of them, the automated system worked fine. It correlated really well. You can see our correlation coefficients were, a lot of times over 0.9. It correlated really well with our manual tree ring measuring. But in other cases, it did not do well at all. And we found that some of these trees growing in the marsh have incredibly small rings. We're talking less than 0.1 millimeters. So these are teeny, teeny, tiny rings. And in some cases, it is interesting. It actually made the wood more dense. We tagged all of these trees, and the ones that are growing nearest to the marsh, the wood in the trees is actually so dense that we couldn't even get the nails to go into the tree. So on these trees with these teeny, teeny, tiny, tiny rings, the automated system couldn't pick up. It couldn't see them. And so this gave us some standards on how we're going forward with measuring, because we need to look at the tree rings first, look at their ring size to determine if an automated measuring will work, or if we're going to have to do that more labor-intensive manual measuring. Also, just a quick look at, because they have collected some data, these are not yet properly cross-dated. With tree ring analysis, 1 or 2 trees, or even 3 or 4 or 5 trees don't really tell us anything, because we know that there are like little microclimates, and maybe a tree is growing just, near a spring, or maybe, some disease broke out and killed the trees around it and it had a lot of light. One tree isn't going to tell us the environmental condition. But when we have 10 trees or 20 trees or 30 trees or more, and we start to see the same signal showing up in all of their rings, then we're going to know that this is representative of some environmental conditions. And we're still in that process. But preliminary data here shows that we kind of see two periods of germination, one happening in the 30s and then another one happening in the sort of 60s and 70s. And this system has not experienced any germination post 1975. So for quite a few decades now, there has not been tree regeneration, mixing two words together there. It hasn't been occurring for quite a while. So once we're completed with this process, we can really go in and look at what was happening at that time frame. You know, were there major storms that came through, or what was the sea level rise, salinity levels looking like? We have currently only processed and measured 45 of these cores, and so we need to, 43, and then we need to go and process the ones that we just collected last Friday. All of those will need to be measured, and then we're going to have to go through the whole quality control cross-dating process. Once that's completed, we will compare our data to climate weather data sets looking at temperature, precipitation, storm events. And once that is done, then we will repeat this process at the VCR and then whatever study sites that we end up selecting in Delaware. This is a 5-year project, and we are just about 10 months into the first year. So we're pretty early in this stage, and we have a lot of work still to go. This research was funded through the Collaborative Research Network, the Coastal Critical Zone Project, and there are a number of students that really helped collect data working on this project, undergraduates being Anthony Baker, Lindsay Koyagni, graduate students Cassandra Miller and Sydney Hall. The collaborators on this grant, they're kind of these sort of superstars that I'm really excited to work with. Holly Michael, Karen Geeden, Kate Tolley, Matt Kerwin, Sergio Fugarzi, Jeanette Miller, Yuping Chen, Angelina Seaforth, Amy Slocum, Yolanda Williams Bay, Rachel McQuiggan, and then also Leanne Hoff from the PDE has come out and helped us collect cores and process the data. So with that, I would like to conclude my presentation and open it up for questions.
Thank you, Stephanie. Really appreciate that presentation. If anybody has a question, please feel free to put it in the chat box. There was a follow-up question too that both of you might want to answer. Jerry asked that they're in a community with a self-managed HOA which is surrounded by watershed forest. Should they advise residents to stop transporting their grass, tree clippings, and other vegetation as refuse into the forest? That there's a suspect of danger of introducing disease in non-native species, etc. So I guess the question is, should they stop putting their grass clippings in the adjacent forest? And so I would suspect that there could be transference there, but Tara or Stephanie, if you have any thoughts.
Yeah, I'm sorry. I just responded back to Jerry. I think angrily by accident in the chat. Yeah, I would suggest not putting yard waste in our forest just because you don't know what you might be transporting. And sometimes we have invaders, like I have some invaders in my backyard that are really hard to get control of, and some people may not know that. And so if they're taking up yard waste and putting it into our forest, it could be potentially problematic. So I would suggest to refrain from doing that.
Great, thank you. And Stephanie, I know that go ahead, I'm sorry.
Sorry, this is Mike. I have a question for Stephanie, if that's okay.
Sure, go right ahead, Mike.
Hi, Stephanie.
Hello. Hi, Mike.
How you been? So in your presentation, you said that you tried to take the core samples all the way through. Do you mean all the way through the other side of the tree or to the pith?
No, well, we always try to get the pith. Because especially when you're trying to get germination. And I know that when you core trees, you like to do perpendicular coring.
Right.
It's always a good idea to get duplicate samples. But with the quantity of cores that we're getting and the size of the team that we have, if we can just core actually going all the way through the tree, then you get a duplicate core in one bore because you'll get coming from both sides.
Okay, yeah, I was just wondering about that. Now, we were coring trees, oaks up to 22 or 24 inches in diameter, so to go all the way through was kind of impossible. So,
and you have to have a huge board to do that. So it depends on the situation. And it also depends on if the tree is leaning or not.
right. So the idea is you just want a duplicate core. Okay,
Yeah.
that answers my question.
Yeah. And then I see in the quote in the box here that somebody was asking about collaborating with the Delaware Geologic Survey, and yes, I have spoken with Todd and also with John Callahan. In fact, I've got a paper coming out in the Journal of Coastal Research with him right now. So yeah, absolutely, we are definitely looking at those collaborations.
Yeah, there was another question. What do you think may have been going on from 1940 to 1965 that might have stalled regeneration?
We need to look into this a little bit more. Interestingly enough, and this is all very preliminary, so I need to really investigate it more. It actually seemed like the 1930 peak was more inland. And then that second peak that was in the 60s was actually more out into the marsh. So the forest was actually expanding out during that time. We did have a very dry, globally dry time kind of in the early 30s. So it could have been related. Yeah, we really just need to investigate more. It is kind of a weird that you have that sort of bimodal distribution.
Thank you. One more question. Would you expect to see species-specific differences in halting of tree regeneration?
It's going to happen at different time frames because some species are more salt tolerant than others. Also, some species are more sensitive to different active layers or your groundwater levels. We did pick loblolly pine here because it is the dominant species. There's a little bit of cedar, there's a little bit of holly. When you get into the higher forests, then you start to see your mixed hardwoods. But yes, the different species are going to see different responses, which is why we are trying to kind of stick with one species for this project. But yeah.
So as a follow-up to that, you're looking for a site in Delaware, correct? Mm-hmm. And are you looking for, so then I'm assuming, does that site need to have loblolly pine associated with it?
We've had to get real with like in an ideal world, yes.
Okay.
It would be loblolly pine, but we are more interested in having this a wider band with this transition. The species is of secondary importance.
Okay, great. That's great to know. So if anybody does know of a site, please get up with Stephanie. I'm sure she'd be happy to have another site here in Delaware. So
yeah, we have tiny coastal forests that we're working with, like some people from Massachusetts, and they're like, why are they so small? I'm like, they just are.
Well, thank you so very much. We really appreciate that. We're going to be moving on to Lizzie Powell. And Lizzie is our final speaker this morning. She's going to be presenting on using airborne light detecting ranging LiDAR to detect changes in the upland forest canopy and the upland marsh border in areas of Delaware. Lizzie.
I need to unmute myself. Okay. Okay, so everyone can see my title slide, correct? Perfect. So hi, I'm Lizzie. I'm a PhD student at University of Maryland studying geographic science. Today I'll be discussing some findings from recent research that I've been doing that's associated with my fellowship in the first chapter of my dissertation, detecting changes in upland forest canopy in the upland marsh border in an area of Delaware using airborne LiDAR. Hold on, I keep on unmuting myself. Okay. So I was awarded the Margaret A. Davidson Fellowship this academic year. The Margaret A. Davidson Graduate Fellowship is a new program that provides funding to graduate students to conduct estuarine research within the 29 National Estuarine Research Reserve systems. I applied to the Delaware NER as I'm interested to see the effect or to understand the effects of sea level rise on coastal ecosystems. So a little bit of background, and Stephanie touched on this a fair amount and talking about ghost forests. So this is like a great transition into what I'm studying as well. So sea level rise rates have tripled in the last 150 years in some areas. And the Mid-Atlantic is experiencing rates of sea level rise 3 to 4 times higher than global averages. This is caused by Gulf Stream changes and land subsidence. But here in the Mid-Atlantic, the coastal plain has very gentle slopes, and it provides a good opportunity for marshes to move up into the upland. This is why we're seeing higher rates of coastal forest dieback than compared to the U.S. Pacific and the New England coasts. Maritime forest decline has been well documented in the southeastern U.S., as well as the Gulf Coast of Florida, North Carolina, and Louisiana. Still piggyback off what Stephanie said, marsh transgression. The rise in sea level leads to the reduction of mainland elevation, which increases the vulnerability to inundation of adjacent uplands. This leads to coastal transgression that was identified by Brinson et al. In 1995. The development of the marsh migration inland from increased inundation and salt stress inhibits forest regeneration and leads to the death of adult trees, termed as ghost forest. And so this is just a little bit of a cartoon here where we have, typical wetland zonation where we have the water marsh edge with mean sea level. We have a healthy salt marsh, some, Phragmites here and there, and then we have an upland forest. With sea level rise, we have the water marsh edge migrates inward, and then you have the horizontal marsh migration going into what used to be upland forest. And so here we have, some dead trees and, ghost forests that surround, a healthy upland forest that could be retreating inward. But sea level rise is not the only factor leading into the creation of ghost forests. Rapid change can occur when upland forests are exposed to flooding due to intense storm surges. That storm surge and associated salination can trigger instant mortality, or the salinity pools can persist for decades and impact long-term tree growth. And this is what we would like to call sea level rise would be referred to a press disturbance, a gradual change over time, while a pulse disturbance is like a pulse of salt water, and that, having a more instant disturbance rather than something that's more gradual. So resiliency, the term resiliency, it was identified by Halling, or ecological resiliency has been identified by Halling in 1973. It refers to the amount of change that ecosystem can endure without having a permanent state change. And coastal resiliency is a red hot topic right now in terms of research interest. But on the perspective of marshes, namely understanding how coastal wetlands will respond to sea level rise and coastal storms. But understanding how the marsh will respond will also help us understand our forestry resilience. For example, will the resiliency of the forest independently hinder the marsh moving into the upland, or will the marsh moving inland reduce the forest resiliency and decrease the forest that's there? And then also, do we see different responses in areas that have been managed with wetland management compared to areas that haven't had wetland management? So for example, Smith et al. In 2017 found that marshes that were subject to freshwater impoundment along the Delaware Bay in New Jersey seemed to be less resilient as marshes that were never impounded for freshwater. And that got me thinking to a more detailed question, or the big question. How will Delaware upland forest respond to sea level rise and coastal storms? Since Delaware coastal areas have been managed throughout history, including straightening, we have a diking situation for freshwater impoundment, and then we have grid ditching for salt hay farming. And I'm wondering how these management techniques will affect upland forests during migration. As I said in the last slide, Smith et al. In 2007 found that impoundments have been associated with decrease of marsh resiliency, namely marsh conversion to open water and increased erosion. And how will that affect forests that border these wetlands, and how will they respond? and specifically how will Delaware upland forests respond to marsh migration and sea level rise in coastal storms, and will actually the historical wetland management affect upland forests during that migration? But I won't be able to answer all these questions fully in this presentation, but I can kind of shed some light on some preliminary findings and plans for the future to answer these questions. So moving right on to the methods. So most studies that study marsh migration use maps, aerial photographs, satellite images to manually digitize and report the change in the transition zone between the upland and marsh over time. This approach has been shown to be useful to trace landward migration, but it could be vulnerable to missing some initial changes to the canopy or understory that might reflect some initial stresses to the canopy. So in the lab that I work at Maryland, they really have been using a lot of LiDAR to understand vegetation dynamics. And airborne LiDAR is a LiDAR system on an airplane that scans over a landscape, and the light energy bounces off the forest canopy. And then the return time of these laser pulses can be converted into a distance, allowing us to map the height of the trees that make up the top of the canopy, and then also and then bounce off the ground, get the height of the ground, and then also any density of the understory that's present there. So with all those returns, it turns into a point cloud with all the returns from the plane acquisition, and then we can use that point cloud to derive specific LiDAR metrics. LiDAR has been proven a great tool for forestry, as the laser pulsar from the system can have the ability to measure not only the canopy, but the density of the vegetation underneath the canopy, which in our case could really show characteristics of upslope marsh migration or the lack of upslope marsh migration from increased forest resiliency to actual marsh migration. So this is going right still into my methods. So I picked a small area that's north of the Delaware, north of the St. John's Reserve, right here right by the Air Force Base. So in the airborne point clouds from 2007 and 2014 were obtained for a small area of Delaware that appears to have undergone rapid transition from forest to marsh. And I wanted to use this area as it could provide a really good case study for some rapid changes that we could be seeing. The point clouds were processed into normalized digital surface models, or the height above the ground with a 1 meter resolution. Then I took the local maximum height of that normalized digital surface model to generate an actual canopy height model. So a raster that with each grid cell is 1 meter, and then it has the max height of the tree in that given 1 meter cell. Then we can observe those height changes within 1 meter from time 1 to time 2. Then I generated a height transition matrix from time 1 to time 2 using methods from Kellner et al. In 2009 and Dubaya et al. In 2009 as well. So here on the top row, we have a aerial image of the study area between 2007 and 2015. And below we have their associated canopy height models in 2007 and 2014. So pay attention to the legend right here. We have some low vegetation in the blue. So we have blue is ground or really pretty short vegetation, where we have green, orange, red, burnt orange, brown, and white are indicative of higher vegetation, so going up to 36 meters. And then in 2014, we see higher instances of that ground signal, where we have pretty short vegetation, or vegetation that is the max height of that grid cell was 0. And it's moving inward here, and then all along the marsh boundary, forest marsh boundary along here as well. So having a more detailed look of what we see. So I took the canopy height model from 2014 and subtracted it from the 2007 canopy height to see the change. And in red, we have the more negative change is indicative of height losses, where in green, we have positive indicating height gains. And so in the red, you can kind of see it's a sporadic, it's kind of almost like a thinning that we see in this, the forest that was the forest before. So specifically along this area here, as well as some pretty intense spots along, and then also right here as well. But, you know, mostly in the pattern of along the boundary, the marsh boundary. And then on the right, we have a distribution of those changes. And as you can see from the distribution, they appear to be left skewed, so reflecting a possible disturbance where we have more instances of negative change rather than a positive change. So going into my transition matrix, this is a little bit confusing, so I'm going to try my best to really try to make it as clear as possible. So transition matrices are really used in land use dynamics and land use change, but they could be used for height changes as well, using LiDAR. So transition matrices can be used to observe dynamic changes for particular height classes from time 1 to time 2. And a transition matrix is used to estimate the probability that the height class at column I will enter a height class that's in row J between time 1 and time 2. So columns for the matrix were interpreted as prospective height changes, or the probability of the height class occupying different height class in the future, where the rows are interpreted as retrospective height changes, so changes that occurred in the past. But if the canopy height distributions for the two time points are similar, we can assume that the study area is in near steady state equilibrium. So this is a lot to digest here, but if we just pay attention to the bolded diagonal are areas that experience no net change in this area for those height classes. But areas that are above the diagonal experience net height losses, where and then below the diagonal we have the net height gains. Specifically looking at the top row, so this is where where we have transitions from 2007 transitioning to 2014. So if we have a height class of the taller height classes are 46% of those height classes are converting to less than 2 meters. And I found that about 20% of all the total land area in that area specifically transitioned from taller trees that converted into less than 2 meters. So really indicative of what we're seeing with marsh migration, where you have tall trees converting into marsh that is less than 2 meters. So so the key takeaways from this small little preliminary study is that we found canopy height changes from 2007 to 2014 using publicly available airborne LiDAR. We found that the most transition is to the 2 meter height class, consistent with the notion of marsh migration. This is also consistent that we saw with the aerial photography for this area. But now when I was doing this, I was wondering, well, what caused this? What could cause this, massive transition over only a 7-year period? So I wanted to look at the water level. So the mean water level for the area this is the mean water level for the area that's close by. And as you can see from the graph, there doesn't really appear to be much of a sea level rise trend in this area, but we do see, some big, massive peaks of the water level, indicating maybe some coastal storms. And so then looking at the Google Earth imagery from 2007, 2011, and 2014, there might have been a breach to this freshwater system, or freshwater impounded system. So the area looks like it was tidally restricted until about 2011, where we see a breach. And this breach could have been caused by Hurricane Irene or Hurricane Sandy that could have made an influx of salt water to the system, killing the trees, indicating more of a pulse disturbance rather than just from sea level rise. So looking ahead, so the preliminary findings for this small area appears to have been impounded, transition from forest to marsh within 7 years. But this begs the question, are there more areas in Delaware that are like this? And do breaches to freshwater impoundments cause sudden marsh migration and mortality of forests in these areas? And are there different areas that and this is different than an area that is more consistently flushed by the tides, like a more open system that we're seeing like at the St. Jones Reserve? I plan to learn more about this and about different management practices in Delaware and see how the forest is responding to increased sea level rise and coastal storms. I plan on extending on this work with fusion between multispectral imagery to characterize forest along the Delaware coast and to identify any patterns with historical management. And also planning on identifying initial changes to the forest structure with salinity with a terrestrial laser scanner, which is another form of LIDAR, but just on the ground. And potentially scaling these findings to looking at other areas like Maryland and New Jersey. And with that, that's all I have for today. And thanks for listening, and I'm glad to take any questions.
Thank you so much, Lizzie. Feel free to put any questions in the chat box. And we are just happy to have Lizzie as part of the Delaware National Estuarine Research Reserve as our fellow. And so we're happy to have her conducting this research in this area. I did have a question for you, Lizzie. Are you going to be looking at sites where there is this possibility for breaches? Are you going to extend it to, I mean, specifically looking for sites where there might be breaches, or what are your thoughts on future sites?
Right now, I've been talking with Carrie about that actually, and so yeah, right now I'm thinking about having 2 areas that are tidally open, so like kind of having, you know, regular, like a traditional tidal flow, and then having 2 areas that are, you know, kind of classically restricted or impounded. But it seemed, yeah, and I have to agree with Stephanie, yeah, it is hard to find a good traditional kind of area that has a good transition zone for the marsh, or marsh to forest, and that's like pretty much the challenge. But I think Delaware could be an interesting site since it is so different. It might behave very differently than what we're, you know, used to seeing in, you know, Maryland and Virginia.
Interesting. Stephanie, you had a question if you wanted to go ahead and come off mute.
Sure. I was just curious about your impoundment because I just about a month ago went to a forest that died. It was along a freshwater impoundment that was breached in 2011, and there was significant dieback. And I was hoping I could use that as a study site. And I went and looked at it, and there are just baby trees everywhere. Apparently, you know, after that breach happened, the system, it's, you know, it's now been 10 years, but this system is really recovering. And I was just curious if you have actually, like, on the ground visited any of the sites that are near freshwater impoundments to see if, like, it really did fully transition, or if, because it was a pulse event, is now recovering.
No, I have not, and that's my plans for this summer. But yeah, I remember you talking about that in the meeting, I guess, the Coast or the Critical Zone Network meeting, and yeah, and I'm interested in that. I'm interested in general seeing, like, if we're seeing regeneration from the pulse events. But I'm wondering how long do they have? Like, are they just living until the next storm and then they die? And so I'm very interested in that, and to see, like, if we can see that with remote sensing, it could be really cool and scalable.
Definitely.
Carrie asked if it wasn't, is that Delaware Wildlands? I'm assuming that's questions for you, Stephanie.
Yeah, it's near Big Stone Beach.
Great, thank you. Any other questions today for any of our panelists? Oops, sorry about the technical difficulty. There we go. Any last minute thoughts? Yes, I agree, Sumner. Thank you so much. We did have some great presentations today. Thank you. A huge thank you to our panelists. Thank you, Tara, Stephanie, and Lizzie. And thank you to everyone who joined us for this webinar today. We will be sending out a follow-up email with a link to the recording and a survey, as we would love to capture your thoughts and feedback about this webinar and the webinar series in general. The next webinar in the series will be held this summer around June or July timeframe as we explore the cultural perspectives of forests and land management. Thank you again for joining us today, and we look forward to seeing you in the summer. Have a great rest of your Friday, and we'll hope to see you in June.
Thank you.
Thank you, everyone.